Hello all, welcome back. So in this video, we will discuss about the second part of the catabolism of fatty acids, which is the beta oxidation. Now in this video, we will be focusing mainly on the beta oxidation of palmitic acid, which is one type of fatty acid, and we will see how the fatty acid, which has now been converted into fatty acyl CoA, is going to be broken down inside the mitochondrial matrix. This is to show you the summary of whatever we have seen in the video. So we are ready with the acyl CoA that has now entered inside our mitochondria, and then now the next step is to oxidize it, start breaking it down to form our final product. So beta oxidation is the last step, but beta oxidation itself is made up of 4 steps underneath it, or you can say that there are 4 reactions that are occurring in a cyclic manner, and that is what beta oxidation is all about. So the 4 steps of beta oxidation are shown over here. The first step is oxidation, followed by hydration. Then we have a third step of again oxidation, followed by the thiolytic cleavage to get our final product. Now these 4 steps, or you can say these 4 reactions, occur continuously or occur in a loop based on what is the size of the fatty acid we are talking about. So at the end of one cycle, at the end of one complete cycle of these 4 steps, the 2 compounds that we get are a molecule of acetyl CoA, which is basically a 2-carbon compound, and we also get a fatty acid chain, which is now 2 carbons shorter than what we started with, than the parent that we started with. So if I started with a 16 carbon fatty acid, what I now get is a 14 carbon acid, and I get one more molecule of acetyl CoA. So based on which fatty acid I'm trying to break down, that many cycles have to be run. How do we decide that? So all the 4 steps are repeated for n divided by 2 minus 1 times. What does n stand over here? N is equal to the number of carbon atoms that are present in that particular fatty acid. For example, let us take the example of palmitic acid, because that is what we will be focusing on. Palmitic acid contains 16 carbon atoms. So the number of cycles it will run is 16 divided by 2 minus 1, which is equal to 7 cycles. There will be 7 cycles of oxidation, hydration, oxidation, thiolytic cleavage, which will ultimately, at the end of it, I will get acetyl CoA. Completely the entire palmitic acid will be broken down into acetyl CoA. I'll be actually getting 8 molecules of acetyl CoA. We'll see how we get this. And then I'll also get some NADH and FADH, some reducing equivalents, which are all going to give me ATP. But what you need to remember here is that based on what the number of carbon atoms is in the fatty acid that we're talking about, 1 less, half of that minus 1 number of cycles will be run. Why minus 1 or why exactly this is happening, we'll see in the upcoming slides. So let's start with each one of these steps in detail. The first step is oxidation of that particular fatty acyl CoA. Now, like I said, we are talking about the palmitic acid. So we'll start with the palmitoyl CoA. Palmitoyl CoA, as you can see over here, is having 16 carbon atoms. You can count the number of carbon atoms that are there on the slide. So I have written 12 carbon atoms together because it's just for the ease of writing. There's one end carbon that I've shown as CH3. Then I've shown you the alpha and beta carbon. Keep a watch on these two because this is where all the action is going to happen. And then we have our functional group in the form of C double bond O, and then finally the thioester linkage, the CoA part. Now this is the palmitoyl CoA. The first step is oxidation of palmitoyl CoA. So we have the palmitoyl CoA, and now as you can see over here, the palmitoyl CoA is getting oxidized. Now this is actually an example of a redox reaction. That means palmitoyl CoA, yes, our friend palmitoyl is getting oxidized, but then something else is getting reduced. So if you see over here, oxidation, we all know, is the loss of electrons. So at the alpha and beta carbon, we have removal of hydrogen. That's why in many books you will also find this step being called as dehydrogenation, because it's actually removal of hydrogen, which is also called as oxidation. And now the alpha and beta carbons develop a double bond. As you can see here, a double bond is developed between these two because they lost one hydrogen. Now that hydrogen which they have lost is being gained by someone else, and that someone else is FAD. So FAD, which is flavin adenine dinucleotide, accepts the hydrogen which is being released by our substrate, that is palmitoyl CoA, and then gets converted into FADH2. This is a very important compound because it's again acting as an energy carrier. It is going to give us ATP later. So FAD gets converted to FADH2. Why? Because it has taken up the hydrogen which was lost by palmitoyl-CoA. Now, one key for you, one easy tip for you all to remember is whenever there is a redox reaction happening or occurring, the enzyme is dehydrogenase. Dehydrogenase is a very common enzyme that is involved in transfer of hydrogen, or in other words, the oxidation reduction reaction. So dehydrogenase enzyme or the acyl-CoA. Why acyl-CoA? Because I'm talking about palmitoyl-CoA. So acyl-CoA, just put the substrate name over there. Think about the action. It's a redox reaction. So the enzyme is dehydrogenase, and whenever there's a dehydrogenase, you need to write a reducing equivalent, which in this case is FAD, that is flavin adenine dinucleotide. FAD gets converted into FADH2. So now when this reaction occurs, I now have development of an enol group, because now the hydrogen has been removed from the alpha and beta carbon, and now what I get over here is called as the trans-enoyl-CoA. This is the first part of the reaction. That is the first step. Out of the 4 steps, we have finished with oxidation of palmitoyl-CoA to trans-enoyl-CoA. The trans-enoyl-CoA now undergoes a process of hydration. Hydration is where you have addition of water molecules. So we need to show that. If we say there is hydration, that has to be shown in this step as well. So we have addition of a water molecule into this reaction, and when that water molecule is added, you can see over here at the alpha and beta carbon, now the double bond is being, you have the removal of the double bond, and at alpha carbon I have a hydrogen being added. The hydrogen came from water. The OH, the hydroxyl part, is being added to the beta carbon. So now I have the formation of hydroxyacyl-CoA. Because this is a hydration reaction, the enzyme name is hydratase. And what was our substrate? Just put the substrate name as prefix. So my substrate name was enoyl-CoA or trans-enoyl-CoA. Hence, my enzyme name is enoyl-CoA hydratase. The work is hydration, hence the name of the enzyme is hydratase. Put the substrate name over there, I get my full enzyme name. So in this step, you have addition of a water molecule, due to which the alpha and beta carbon now have accepted a hydrogen and the hydroxyl part to form the final molecule that is being formed over here is the hydroxyacyl-CoA. This was at the end of the second step or the second reaction. The third reaction is where this hydroxyacyl-CoA gives rise to ketoacyl-CoA. Now what happens over here? Look at the third step. So hydroxyacyl-CoA is now going to undergo one more round of oxidation. I've already told you this. When there's oxidation and there's redox reaction that's occurring, the name of the enzyme is dehydrogenase. You can always write that. And when there's a dehydrogenase enzyme, if this hydroxyacyl-CoA is getting oxidized, then something else has to get reduced. It has to happen. So you see over here at the alpha and beta carbon, I now have the beta carbon losing the hydrogen. See over here, the beta carbon has lost the hydrogen. Only one hydrogen is being lost. But if this is losing the hydrogen, someone else has to catch it. So the molecule that catches the hydrogen or that accepts the hydrogen over here is another very important reducing equivalent, that is NAD. NAD stands for nicotinamide adenine dinucleotide. Nicotinamide adenine dinucleotide takes up that particular hydrogen to form NADH. So I have NAD accepting that particular hydrogen which has been lost by the hydroxyacyl-CoA to form NADH. Now this is again, the enzyme name is easy for you all to write. It's dehydrogenase because you have a redox reaction that's occurring over here, and the substrate name you need to put it over there. So the enzyme name, the complete enzyme name is hydroxyacyl-CoA dehydrogenase. So now we have finished with 3 steps: oxidation, hydration, and another round of oxidation. Now coming to the last step. In the last step, we have the ketoacyl-CoA which was formed in the previous step undergoing thiolysis. Thiolysis is basically, so thio is some prefix that we use usually for anything related to sulfur, and lysis, like we all know, is breakdown. So we are finally cutting down that molecule. We are finally going to, this is now a balanced molecule that has been formed. We are going to break it down with the addition of a thio compound, or with the addition of a compound that contains sulfur. Now the compound that contains sulfur, which is going to be added over here, is the CoA or coenzyme A. So what we do over here is we are going to add one more molecule of coenzyme A. Let me just write it over here. We are going to add one more molecule of coenzyme A, which is often written like this. So coenzyme A is being added into the reaction mixture, because when that gets added, now the final cut, this is the whole reason why we call it as beta oxidation. The cut occurs over here between the alpha and the beta carbon. One CoA is being kept by the acetyl CoA. So you now have two compounds being formed over here. One CoA, which was already there in the parent molecule, is being kept by acetyl CoA, a 2-carbon compound. And this new CoA that we added is being added at this end, at the beta carbon, to now form a fatty acid, which is 2 carbons shorter than the parent molecule. So we started with a 16-carbon palmitoyl CoA, and now the molecule that we get over here is having 14 carbons. You can count it. It is having 14 carbons, and this is called as myristoyl CoA. So now myristoyl CoA is being formed, and I have one acetyl CoA that has been produced. But we don't stop over here. I want only acetyl CoA. I don't have any function of these intermediates. So now myristoyl CoA will get back into the cycle. When I say get back into the cycle, what happens over there? The myristoyl CoA is now going to go and start with the oxidation part right over here. So myristoyl CoA starts with oxidation, then hydration, then oxidation, then thiolysis to give me another molecule. So let's see what happens over there. So this was the summary that, or you can, this is what you can write for your exam as well. We have palmitoyl CoA, which has now given us the, it has formed the trans-enoyl CoA after the first step, which has then given me the hydroxyacyl CoA, that has given me the ketoacyl CoA, and finally that has given us acetyl CoA and a 2-carbon lesser compound, a 14-carbon fatty acid, that is called as the myristoyl CoA. So we started with a 16-carbon compound, and that gave us a 14-carbon compound after one round of beta oxidation along with the acetyl CoA, which is a 2-carbon compound. Now this 14-carbon compound, that is myristoyl CoA, it doesn't stop over there. This 14-carbon compound is now going to enter into several more cycles of the same 4 steps. So myristoyl CoA undergoes oxidation, hydration, oxidation, thiolysis to give us a 12-carbon compound, that is lauryl CoA. And then lauryl CoA undergoes the same 4 steps to give us decanoyl CoA. That again enters into the same 4 steps to give us octanoyl CoA, which gives us hexanoyl CoA, butanol CoA, and acetyl CoA. Now at the end of 7 cycles, why have I said 7 cycles? The first cycle is when we have the palmitoyl CoA breaking down to myristoyl, that is 1. 2 is here, 3, 4, 5, 6, 7. So we have 7 cycles of this running for us to get at the end of it 8 molecules of acetyl CoA. How is that coming about? So 8 molecules of acetyl CoA, 7 I get at each cycle. We are running 7 cycles of this. So I get 1 at each cycle. For example, I get 1 over here, then I get 1 over here. When lauryl CoA is getting broken down, I get here, I get here. At every step, I get 1 acetyl CoA. But then when butanoyl CoA breaks down, butanol CoA is a 4-carbon compound. So when 4 breaks down, I actually get 2 molecules of acetyl CoA. That is why at the end of it, we have 8 different or 8 molecules of acetyl CoA in our hand. Now these 8 molecules of acetyl CoA along with the NADH and FADH2 are going to give us a lot of energy. How is that happening? So 8 molecules of acetyl CoA enter into the TCA cycle. You can see the video on TCA cycle or the Krebs cycle over here. When they enter into the TCA cycle, each molecule of acetyl CoA gives us 10 ATPs. How does that come about? Because in the TCA cycle, we get 1 ATP directly from the substrate level phosphorylation. Then we get NADH and FADH2 during TCA cycle. Each of that, both of that together gives us 9 ATP. So together, I get 10 ATP just from 1 acetyl CoA. So you can imagine 1 acetyl CoA itself is giving me such a large amount of ATP. Now I am having 8 acetyl CoA. So 8 into 10, here we get 80 ATP molecules. Each NADH, generally also, whether it is from the TCA or whether it has come from beta oxidation, each NADH can give us 2.5 ATP. Now I'm sure there is a lot of debate about this. Maybe you might have heard of, your teachers might have told, or you might have read about each NADH giving us 3 ATP. But then there are reports, I mean it was 2.5, they had just rounded it off. You need to take that each NADH gives us 2.5 ATP. Again here you can watch the video on oxidative phosphorylation to get clarified about this. So each NADH gives us 2.5 ATP. We have 7 NADH and each FADH2 gives us 1.5 ATP. Here again you might see a lot of books giving you the whole number as 2 ATP. But together these 7 NADH and 7 FADH2 together, they enter the electron transport chain and give us 28 ATP. So I get 28 ATP from here. I get 80 ATP from acetyl-CoA, that is including the TCA cycle and the electron transport chain. Together I am going to get 106 ATP. But remember one thing, actually I get 108 ATP. But remember one thing, we used 2 ATPs in the activation of the fatty acid. Now you may tell me we had added only 1 ATP molecule over there. You're right. But then that 1 ATP molecule was, in that 1 ATP molecule we had broken down 2 high energy bonds, because from ATP we had got AMP, if you remember. So ATP had given us AMP. That means I have used up 2 high energy bonds, which is the equivalent of 2 ATP. So we subtract 2 over here for the activation of the fatty acid, and now the net ATP that we get over here is 106. This is what you get. 106 ATP is the net energy yield from one fatty acid molecule, from one palmitic acid molecule. Now in several books or papers or online you would find it as 129 ATP. That is again coming because of the assumption that one NADH gives you 3 ATP and one FADH2 gives you 2. That is why they have got 129. But the actual number of ATP that we get from one palmitic acid is 106 ATP. So this was the energy yield from beta oxidation. Like you see, it's much more than what I would get by breakdown of one glucose molecule. That is why this is considered, lipids are considered to be an energy storage molecule as well. Because when we really require, for example, in hibernating animals, when they really require the energy source, lipids is what acts as the major energy source, because they give us a very high quantity of energy, very high yield of ATP in comparison to a carbohydrate molecule being broken down. I hope this video was useful to all of you, and I do wish to see you all in the next ones as well. Thank you.